for the invitation. I'm very grateful to have the opportunity to tell you a little bit about what we are doing here in my lab. Yeah, as Michael mentioned, I'm from the School of Mechanical Engineering in Tel Aviv University. And I thought to start maybe with a brief overview on my lab. So basically, we're doing a lot of experiments at the single cell level, how cells interact with their extracellular matrix. It's basically a fibrillar matrix made of different proteins like collagen and fibrin. So we look a lot about the mechanics of fiber networks and how cells mechanically interact, how the forces that cells apply on the matrix, how they propagate, and so on. I will tell you more about it in a minute. But I also would like to mention that we're also doing some work on tissue mechanics. So this is more like when we have many cells, we try to grow maybe tissues, but we always use the knowledge from the cell matrix mechanics to take to the tissue level in order to create better tissues, more functional tissues, and so on. And here we did a lot of work mostly on skeletal muscle tissue. So this is what you see here in green. Do you see my mouse? You see my mouse?
Yes.
Okay.
Yes.
And now we're also doing some work on the iris, which is also a nice tissue made of muscle tissue with different organizations. But I'm not going to talk about it today. Today, I'm more going to focus on the cell matrix mechanics, hopefully, and a little bit, hopefully, about collective behavior that I'm hoping to take more in the future. And I would be very happy to get feedback from you about that. And feel free to ask any question that you may have during the talk. Okay. So the motivation for probably everything that we do in my lab, I think is nicely visualized in this movie. So this is why usually I like to start with that movie. It's taken from this paper. And here, basically, what you see is a fibroblast cell. Everything that we do is in vitro. Okay, I know that you're doing a lot of in vivo work. We are not working with animals. We like to have in vitro model systems that are very much controlled and try to get understanding of those systems. So here you can see this is a reconstituted collagen gel. The fibers of the gel are fluorescently labeled, so you can see them in green. And this is just the fibroblast cell labeled in pink with its nucleus. And I will run the movie, and what you can see first is a little bit about the 3D view of the system. But then you can see that the cell is migrating, and when it's migrating, it's really pushing and pulling on the surrounding fibers, creating a lot of movements around it. And those movements are because the cell is pulling on the fibers. So basically the cell is a little bit like a biological machine. There is a lot of mechanics here. The cells apply active forces on the surrounding ECM using the actin myosin machinery. Okay, and the cell is basically adhered to the fiber, so it's mechanically coupled to the fibers. So the forces that the cells apply using its actin myosin machinery, the forces basically are transmitted to the environment, creating a lot of displacements, fields, stresses, and so on around the cell, even farther away from the cell. And this is what I hope you can clearly see in this movie. So for us, it's a great mechanical system. First, because the cells apply forces, and second is because this ECM environment, or this reconstituted collagen gel, in this case, has actually very interesting mechanical properties. First, is very often 3D. It's very soft gel. It's made of fibers, which give rise to very interesting mechanical effects. It's highly viscoelastic and non-linear, which I'm not going to get in depth, but the nonlinear properties are actually quite important. I will maybe mention it a little bit later on. And those forces that cells generate actually are important in many different biological functions. As you can see here, for cells to migrate in the mesenchymal mode, they need to apply forces in order to push themselves forward. So this is one function, but there are many different functions like cell division and cell differentiation, in which the forces are very important. But I will mention that my lab has been mostly interested in whether those forces can be used by cells to communicate with each other. And here I would like to talk a little bit about that aspect. I want to move this Zoom bar because it's interfering. Okay, hide. Okay, great. So the concept of mechanical interaction between cells is shown here. So the cells can apply forces. So those are mainly contractile forces because the actin myosin machinery are basically contractile. And those forces can be transmitted to the ECM surrounding, as you just saw in the movie, reaching nearby cells. And those forces may affect their function. So in a way, this can be used for cells to communicate in a long range, not in a direct communication, but indirectly through the ECM fibers. So that's the idea of long range mechanical interaction between cells. And I will also mention that the conventional way for communication is through biochemical signals, right? Cells can secrete molecules, and those molecules can diffuse through the ECM and reaching other cells and affecting their behavior and function and so on. So we are talking about communication through mechanical means, mechanical signals. It can be in a way seen in an analogy to a tug of war behavior when two people are pulling on a rope, so they can sense each other through the tension along the rope. This mode of communication, in principle, can be much faster, maybe a longer range than the conventional biochemical communication. Okay. So this is an image which I think capture the different effects that we see in the lab. So this is actually an experiment that we often run in our lab. You can see here in green fibroblast cells, which are highly contractile cells. And in this case, they are seeded in a fluorescently labeled fibrin gel. The fibers are fluorescently labeled, so we can visualize them. And this is just a confocal slice, so it's a cross-section for the gel at some region. And this is a few hours after cell seeding. And what you can see is that the cells are doing a lot of things to the gel fibers. Okay, so they remodeled because of their contractile activity, they remodeled the gel. And you can see a lot of things going on. So you can see fibers that are getting aligned in the area of the cell. You can see densification of fibers, alignment of fibers, compaction of fibers. And also, quite interestingly, you can see that when the cells are at some distances to each other, they also form some connections for those fibers. So the fibers become very tense and aligned in between those contractile cells. And they also, the force that the cells apply can also transmitted through linear-like tracks, as you can see here, which are called force chains. So in this system, because this system is made of fiber networks, which also have those fiber networks have very nonlinear properties, including they tend to buckle in compression, and under tension they tend to stiffen. Okay, so this is the typical behavior of long and slender elements that under compression they buckle and they resist tension. So those different effects at the individual fiber level create a system that faults tend to propagate along specific tracks, which are called fault chain. As you can see here, for example, you can see a long fault chain and also here fault chain. So the fault is not distributed homogeneously, but along specific tracks. And giving rise to these different effects that you see in this image. Okay? And we are...
Sorry, can I ask a question?
Yes, sure. Go ahead.
So these fault chains, do they get strengthened over time, and do they modulate the intercellular communication over time, like some sort of synaptic strengthening or something analogous to that? Do you see anything like that?
If they get strengthened over time, if they get like if we see them more over time, if they get stronger over time, that's the...
Yeah, exactly.
Yes, definitely. So this is actually at very early time point, just within probably like 2 or 3 hours from cell seeding. The cells are still in their spherical shape. Over time, they will spread more. They will start also to degrade the matrix, to secrete their own matrix, and different effects that are still not at that early time. But even though at that early time, they already remodel quite significantly the extracellular matrix. And those fiber chains can connect between nearby cells. And what we try to understand whether they use for cellular interactions. Exactly.
So they conduct voltage. That's what you're saying.
So that's a good question because we haven't got into the bioelectrical properties of those, but it's definitely something interesting to explore. We look at the mechanical properties. So whether what is the stiffness of those elements and how cells respond to those elements. But we haven't looked at the ability to propagate voltage or anything like that.
Because if their thickness change, that can influence on the resistance, which is conduct more or conduct less electricity. Or I don't know if, again, they actually conduct something.
Yeah, it's a good question. I don't really know, but yeah, it's definitely interesting. And your feedback on that, because you have a lot of expertise on that, so it will be interesting maybe to study that. I'm not sure even about the conductivity of those materials. In this case, this is fibrin, but also collagen we use a lot. But yeah, you're right. It's a very interesting direction to explore.
I mean, even in the absence of, even without the bioelectricity, just the biomechanical part. So Richard Watson and Chris Buckley and others have made models. I think this is maybe what Santosh was getting at. Models of collections of springs that do a kind of learning. So they basically simulate the sort of Hebbian kind of thing, but just from the mechanical aspects, right? No electrical. So I think it sounds to me like there's at least 2 layers here where you could find something like the synaptic plasticity that Santosh was referring to. There's the purely mechanical part, and then also on top of that, there may be electrical aspects.
Yes, definitely. We only look at the mechanical part, and throughout the talk, I'm going to mention more and more mechanical details about those remodeling tracks that cells remodel and form in the matrix. But the bioelectrical, properties are, could be very interesting to explore. They're definitely relevant here. Could be relevant.
And just one last note connecting them. So even if there isn't, bioelectric communication at this distance, which I'm not sure that the ECM by itself could, because it doesn't have a cell membrane. There's definitely piezo channels all over the place, right? I mean, that's how we hear and feel and stuff like that.
Yeah.
So just pulling on a cell, I would absolutely expect to have a very big bioelectric effect by a pretty well-known mechanism. So that's super, super cool.
Yeah, that's definitely something that we try to understand better, like how cells respond to those interactions that they form through the matrix. Also, like calcium imaging, that this is something that we would like to do. We still haven't done that. How cells, biologically respond to the mechanical effect that they, by themselves, they generate.
Can I ask a follow-up question? Maybe you will mention that later, but just wondering how the force decay through distance. Like, is it decay like in second order or third order?
Yeah, great question. I have a slide about that. So
Okay, thank you.
wait a little bit and I will get that. Okay. So that's the so that's, I would say, the main subject that my lab has been focusing on in the last several years, about those structures in the matrix that cells create and how they affect their function. So we often see that both in fibrin and collagen matrices, that cells, contractile cells, it doesn't have to be fibroblasts, but also other contractile cells like cancer cells and other types of cells. They create those what we call bands or bridges in the matrix that connect between them. The formation of those bands are directly relevant to the nonlinear properties of the matrix, because we have a lot of buckling of fibers in the transverse direction. And the strain stiffening, so the fact that those fibers get stiffened when they are pulled also is relevant in forming those structures. The distance that cells can interact with those structures can be about 10 times the cell size, as you can see here. So this is a little bit about the range. How cells respond to the formation of those structures that they generate by contracting, I will discuss a little bit later. But essentially what you see, that cells migrate, they can show the shape, they can invade along those structures. And another important aspect is the feedback. That cells, they have a feedback mechanism, obviously, of course, but because once they feel a force in a certain direction, they can increase the contractility. And this can increase the fact that we see those very intense structures forming between cells, because once they form it, it can amplify the formation of those structures because of the feedback mechanism. Okay, just a little bit more about those bands that cells generate in between them. It's a highly dynamic process. It's not always predicted, so not all cell pairs form those structures. It's not only about the distance, it's about the contractile activity, and it can be affected by neighboring cells. For example, you can see in this movie, we have 3 cells this time. The matrix is in red. Those are 3 fibroblasts, and when they spread and interact with the matrix, you can see the formation, you can see some mechanical interaction here and also here. But for some reason that we don't really understand, this cell pair didn't form any interaction. So it's a very interesting question to understand, and we always still trying to understand why when cells are doing that and why they're not always doing that. And this also hint to maybe to collective effects, because if we have many cells, the presence of other cells can affect the interaction. And this I will talk a little bit more in a minute, but I'd also like to mention that not only individual cells are actually doing those mechanical interactions for the matrix, but we also see a similar effect in other more complicated organisms. For example, this is a collaboration with a group in Barcelona, and here they are growing embryos, mouse embryos in collagen gels, and they image both the collagen and the embryos. They try to understand the development of the embryos and the forces that actually affect the development. But if we have 2 embryos at some distance, because of the contractile activity on the collagen, we see actually very similar effects. Okay, so you can see this densification and alignment of fibers in between the embryos. We can actually quantify using image analysis algorithms. We can follow the movement of the fibers, and we can get the displacement field, and based on that, infer some of the forces that are propagated in the matrix. So it looks like that. And this effect, this effect that cells apply some local force, which can actually propagate to relatively long distance along a linear tracks, is actually captured also in synthetic materials, completely out of the biological world. This is, for example, just a ball that my daughter got a few weeks ago. And surprisingly, when we touch the ball with our finger, we really see a nice propagation of forces that propagate in a similar way to our biological materials. This is just a ball that is made of a sponge, so it's quite soft. And the important thing is that it is coated with a very thin plastic coating. And because it's so thin and it's coupled to a very soft material, when we apply a local force, we have a lot of buckling. So it's a very similar nonlinear properties as we have with fiber networks. We have buckling that causing the force to propagate along linear tracks. So you can see it here. So when we press with our finger, you can see how the force propagates along linear tracks. So it's not homogeneous. It has specific tracks that it prefers to propagate along. So this is a very similar effect to what we see in fiber networks. When cells apply a force, it will propagate along linear tracks called force chains. And now if we have two such local forces, if we press with two fingers, we can see the force propagation between the sides of that we applied the force. So this is a very similar effect to what cells are doing. And the bands or the bridges that they form in the matrix in between them because of these contractile forces. Pressing on the ball with our finger, in a way, is very similar to applying a contractile force. And if we do that, and now we add another pair of interactions, we can actually affect the interaction that we had originally. So let me play that again. We have this interaction, and now if we apply two forces here, we actually cancel this interaction, forming new interaction here. So it's, and that's hint again into collective effects, that if we have many cells, the interaction between the cells, they can affect the interaction of the other cells collectively. So this is what we are actually trying to understand better about if we have many cells interacting, and I will tell you a little bit about that also. So just to summarize maybe our main question within this system, our key relevant questions are first about the range of forces. So if we apply a local force, similar to what cells actually apply, what is the range? What is the decay pattern in fibrous materials? Second, how cells respond to the forces or the deformation tracks generated by other nearby cells. So this is a way for cell, this relates to the communication between cells. And eventually, we would like to understand how cells interact collectively in a group and how that may lead to effect on tissue organization and pattern formation. So it's the collective effect is summarized here. If we have a single cell that applies forces, and now we move to a multicellular system. We have many cells, many contractile cells. They can transmit forces in between them, and that can eventually may lead to some interesting collective behavior that affect the organization and pattern formation. And in terms of physiological relevance, we believe it's mainly relevant to tissues in the body that are rich with ECM, and cells are basically more separated from each other. So their interaction is through the matrix and not directly. So, and that could happen this is more relevant to connective tissues, to wound healing, or to fibrosis, where we have a lot of collagen And we have myofibroblasts come in and apply contraction, for example, in fibrosis. Okay, so this is a little bit about our computational. We use a lot of computational modeling to understand how forces are transmitted in fiber networks. So this is just one example out of many of our models. Most of my talk will be about our experiments. So I think this is the only slide about the computational simulations. So in the modeling, we can model the network, the fiber networks. We control everything about it, the geometry, the structure, and the mechanics. And for example, we apply contraction here. So this is a very simplified model of cell contraction. So it looks like that. So basically, we don't really care about the inside of the cell. We just like to understand what's going on in the matrix when cells contract. So you can see when the cell contracts to about, in this case, about 50%, we can see a lot of alignment of fibers in the radial direction in a similar way that we see in experiments. Here we use, by the way, Abacus software. It's a finite element software. And then we can get the displacement fields, the stresses in each individual fiber. So it's discrete finite element simulations. And then we can ask a lot of questions about how forces, for example, in this work, how forces decay and the distance that they can propagate. So for example, here we used different networks with different mechanical properties. For example, we compared between fibers that are linear, so they basically behave as springs, to fibers that stiffen in tension. So when they are under tension, they get stiffer and stiffer. We have a model that we compare to fibers that buckle in compression, and models that we have both, which more realistic model of buckling and stiffening, and that way we can decouple different mechanical effects and start to understand how each mechanical property may affect the propagation. And this is what we see, for example. So this is a quantification of the displacements over distance. It's all normalized. So basically, we look at a radial distance, and we look how the displacements, which are very high near the cell, and they decay over distance, but how they decay for different mechanical model. So as you can see here, this is the linear model, and this is the logarithmic scale. The decay is basically the inverse of the distance with a slope of -1. And this is the theoretical prediction for contraction inclusion in a linear elastic material. But when we start to introduce the nonlinearity, the effect of buckling of fibers and the stiffening of fibers, we get a different profile, which are the orange and the purple lines. So we see that the propagation gets, so the curves gets higher, meaning that the forces basically propagate to a longer distance. So the decay is slower and they reach longer distance. And this is exactly the effect of fiber network, that when forces propagate along fiber network, they can get to much larger distances compared to linear elastic materials. So that was a little bit about our simulations. Another thing that we tried to do was to do that in experiment to try to understand the propagation of forces in those fibrillar materials, but in a very controlled experimental system. Okay, still without any cells. I will get to cells in a minute, but this is just a gel, a three-dimensional gel. In this case, this is probably fibrin. So you can't see the fibers because it's not in a fluorescent. This is just bright field. And here we used optical tweezers. So it's a great system to manipulate objects with the power of light. So it's a collaboration with Raya Sorkin from chemistry here in Tel Aviv University. Okay, sorry. And here what we did was to take the gel, to embed it with small particles, that you can see here. So we actually have a bigger one in the center and a smaller one that distributed around, and they actually stick to the fibers that you can see in this system. And with the optical tweezer, we trap this bead in the center, and we can oscillate it in different directions. This is another nice thing about this system. And then, so we apply a very controlled force. We control the magnitude, the frequency, the direction, and then we try to look on the other beads, how they move around. So it looks like that. So you can see when we move this one, all of the other moves because they're all connected for the fibers, okay? And we can also do that in the transverse direction. And then we can quantify the movement of the beads and to get better understand how forces propagate in this material. This is how the fibers look like. So this is the particle that we oscillate, and this is the cross section of the fibers. So this is an example of how the displacement map looks like. So here we quantify each individual bead around it and quantify the displacement in the axial and in the transverse, in the vertical direction. And we did also another thing within the system. We took the gel and we wanted to measure the gel that is under tension. And the motivation is because many of our tissues are stretched and the fibers aligned in an anisotropic manner. And also this is what cells are doing. As I showed you before, they stretch the fibers. So we want to understand how forces are also transmitted in stretched gels that the fibers are aligned, where the fibers are aligned. So we designed this system to stretch gels. The gel is actually polymerized in this small circle here. And this setup can be placed in the optical tweezers or in the confocal microscope, and we can stretch the gels during the experiment. This is the gel here. This is how it looks like under stretch. So you can see the fibers aligned. So all the properties of the gel are actually changing when we stretch it. They're becoming highly anisotropic. So the direction matters, basically. We get different mechanical properties in different directions. We can also do that in real time. So we can stretch the gel to different levels. And for each stretching magnitude, we can try to understand the response of the gel to the manipulation. So this is how it looks like. So it looks a little bit differently because now we overlay a lot of different regions. So we get the average response of many different regions and many different gels to get more statistics, but it's the same displacement map that I showed before. And in this case, we apply the force in the x-axis, so the force is applied. So we oscillate the particle in the direction of alignment. So the gel is stretched along this direction, x-axis, and to different levels. And this is basically the propagation field. And you can see how it's changing as we stretch the gel more and more. Basically, the propagation becomes more directed in the direction of stretch. So if we have alignment of fibers, we get more propagation along this direction of the fibers. We can do other things, so we can apply the manipulation in the perpendicular direction to the fiber alignment. So the gel is stretched in the x-axis, but we apply the manipulation in the perpendicular direction, and we look how forces propagate. So it looks like that when we increase the stretch, when we have more fiber alignment, the field is changing a little bit. Basically, the force propagate less in the y direction and more in the x direction. And that's actually quite interesting observation that we still do not completely understand, but I'm not going to get into that today. Another thing that we can do with the optical tweezers is not just manipulating objects and see the response of the material, but actually also to measure things. For example, we can measure stiffness, local stiffness. There's a lot of theory about that I'm not going to get into, but we really wanted to measure those structure that cells generate and to measure the stiffness of those structures. Because we have a lot of fiber alignment and densification, those regions are probably much stiffer. And stiffness for cells is actually very important mechanical cue to which cells can respond to. So, for example, we use the optical tweezer also to trap those particles, and by looking at how they, when we oscillate them a little bit and look how they move, we can infer the stiffness in different directions. So you can see some of the values of the stiffness. This area is much stiffer, order of magnitude stiffer than the background matrix, and both axes actually get stiffer. But the axis a lot between the cells, it's becoming much more stiff, and we quantify all of that is shown here. Okay, so I really want to get into our experiment with cells. So that was a little bit about our computational modeling and about experiments with our cells, trying to understand the mechanical properties of those fibrillar gels. But eventually, we want to understand how cells interact with those matrices and in between them. And not only two cells, but we really try to understand a multicellular system. What's happening when we have groups of cells. So so one of the things that we can see, so actually this is a movie, so you can see a little bit over time. This is a confocal real-time imaging. It's actually quite challenging to take those movies. Cells don't really like to do that, to do those interactions under the microscope. They don't really like the laser too much. So this is one of the only actually movies that we have so far, but we have snapshots in time that I will show you. But I do want to mention a little bit about this system, because as you can see here, the cells are quite organized, okay? Because when we put the cells in the gel, they just randomly distributed. So how can we really wanted to make a very control system that we can really position cells in very specific configuration, and we can control the spacing between cells and how many neighbors will be around each cell. And that will be a very controlled system to start to analyze how the interactions between the cells are taking place. You can go to that paper, but basically we start with a 2D pattern on a cover slip. We start with creating adhesive pattern on a cover slip. Here we use like a mask from the clean room and a UV light that goes through the mask, creating adhesive pattern.
Thank you.
Yeah? Question? Okay. We add the cells starts to adhere to the specific adhesive pattern. And then after we get the pattern on the cover slip, basically we add the gel of interest. So it could be fiber, it could be collagen, it could be other type of gel. And we polymer the gel on top of the cells. And then we found a way to peel off the gel with this silicone strip to which the gel adhered to. And we found a good way to peel off the gel, and the cells actually transfer from the 2D glass or cover slip to the gel. So the cells are in pink in this cartoon. So eventually we get a gel with a specific pattern of cells sitting on the top surface of the gel. And we control a lot of things. So we control the size of this adhesive spot. So it can go from individual cells to cell aggregates of different sizes. We control the distances, whether it's a square or hexagonal and so on, okay? And this is how it looks like in real time, in real life. So this is the gel, and this is the silicone strip around the gel. Okay, so one of the first thing that we did was to pattern different types of cells, contractile cells. And this is, for example, specific cancer cells, epithelial cells. It's HRAS transfected cells that we received from a different lab here. And you can see the initial pattern, and you can see what's happening after 6 hours. So those cells are actually highly contractile. So they apply lots of forces, but they maintain in their original aggregate. So this is actually an aggregate of many cells in it. So it's not individual cells. And you can see the same bending behavior that cells form with their neighboring cells. And we start to get a collective system in which cells transform from being isolated from each other to being mechanically connected. And now we want to understand better how this may affect their biological function. So this is actually where we are now. For example, one of the things we wanted to do is calcium imaging, look whether we have some synchronization that is happening because the cells starts to mechanically couple and so on. If you have ideas, I'm very happy to hear. But we are basically at that stage that we see these multicellular interacting systems connected through those bands in the matrix. Here?
I don't know, but I will send him the recording later.
Okay, so this is a higher zoom. You can see that when cells already migrated out, so this is after 6 hours, and this is actin GFP signal, okay? So you can see the cells spread out, but they move along their neighboring aggregates. So they know it's not a random movement. They have preferred direction toward the neighboring cell, toward the neighboring aggregates. And if we look a little bit more into the actin staining, you can really see the very nice stress fibers when cells are along this band, that, you know, this region of highly aligned fibers that cells generate. And the actin signal inside the aggregate is very diffusive. So they really change the way the actin, the morphology of the actin once they go out along this structure in the matrix that is very aligned, very dense, and very stiff, because we measure the stiffness, remember? So it's very stiff material, and the cells definitely respond to that by aligning and configuring their actin machinery. We change the spacing. We try to look at how the spacing affect this behavior, and we mainly see this preferential migration when spacing is about 200 microns in this case, and this preferential migration is lost when we increase the spacing. So this is really response to the effect that we have the neighboring aggregates and the formation of the remodeling tracks in between the cells. This is just more images showing the system. So this is after 3 hours. So cells just start to leave the spheroid. You can see a lot of remodeling of the matrix at this stage. You can see the alignment, and cells starts to respond, and they start to invade out of the spheroid along those structures in the matrix. This is a more advanced time. This is about 9 hours, so we can already see some pattern formation, I think. So cells really move along the formation and tracks, remodeling track that they form in the matrix. This is integrin staining. You can see a lot of integrin staining here. This is just more images showing the remodeling and the response of cells to that remodeling in the matrix. So with that, I would like to finish. This is just a summary slide. We start from here. Basically, we see the mechanical interaction between cells, which can extend to multicellular systems. There could be a lot of signals going on here. We basically focus on the mechanical signals, but there could be biochemical signals and definitely also bioelectrical signals. And from here, whether we can whether this affect pattern formation, this is where we are heading. This is where we would like to understand more. We think we have a great system to explore that, a very controlled system, and we hope to look at the different signals that may take place during that process. So yeah, I would like to thank my great team. Oren, my lab manager, I mainly showed the work of Shachar today, and also Bar, that is a previous member of the group, and our financial support. And thank you for listening. I will take any questions that you may have. Thank you.